Our interests are ranging on flaviviruses and also HIV from the past work. And we are ranging from molecular studies on the virus-host interactions and some applied studies towards the definition of diagnostic tools for flaviviral infection. And Flavivirus is a very large family of important human pathogens that include dengue virus, include West Nile virus, Japanese encephalitis, yellow fever, and also Hep C. Hep C is also a member of the Flaviviridae family. With respect to hepatitis C, which is not delivered by arthropods or by insects, the other members of the family are derived by either mosquitoes or other insects such as ticks in the case of tick-borne encephalitis. And these viruses are distributed worldwide and mostly originate from Africa, and they conquer new areas in time. There's for example, this shown here is the expansion of West Nile virus in the late 90s towards the US. As you may well know, there has been the first cases were in New York, and then they spread west, and now they're spreading south. And now all the continent is also endemic for West Nile virus. And the situation is very complex. There are several different viruses overlapping in areas in Africa, in Asia, and so on and so forth. We conducted a study together with a collaborator in northeast Nigeria, and we wanted to understand what was the seroprevalence of arboviruses in northeast Nigeria. So we surveyed a number of sera from patients returning to the hospital for febrile symptoms, and they were diagnosed as either for malaria typhoid or dengue virus, chikungunya virus, which is not a flavivirus, West Nile virus, and yellow fever virus. And as you can see here, the picture is quite variegated with a lot of co-infection between different flaviviruses. This may be trivial, but it was the first survey in this area since the 60s concerning this kind of viral infection. And with this collaboration, we established in the area the tools for viral diagnosis for flaviviral infections, including serology, molecular biology, and virus growth. Unfortunately, this area is highly troubled, and the person who were in charge of doing this assay had to leave. So it's not possible to do this work over there. And so we were kind of concerned about how to address viral diagnosis in areas where you cannot have a centralized diagnostic laboratory. And so we turned our attentions towards point-of-care devices for diagnostic viral infection. So the concept is quite simple. Nowadays, what we normally do is to have an infection, send the samples to the hospital, get a diagnosis, and get a feedback to the infection. This can take days in places where the facilities are centralized and are nearby the site of an outbreak. However, it becomes highly complicated in areas where facilities are not present, and the response can take weeks. And so what happened, for example, for the Ebola epidemic recently, leaving outbreaks uncontrolled for a long time causes massive problems. The concept of point of care is that you can actually reduce the time from detection to intervention by diagnosing the viruses, also the pathogens, locally. Now, what has been used so far is strip tests. So I'm showing here strip tests. These are the one developed at ICGEB New Delhi. This is a strip test for dengue viral infection and allows you to have serology and antigen detection of dengue virus. However, this is very advantageous. It's very cheap. It costs around $2, but it's monoparametric. It gives you also an answer about dengue virus. If you have a situation where you have co-infection between different pathogens, including other pathogens like malaria, typhoid, in addition to viral infection, you have a problem. You have to have multiparametric assays at the site of infection. So we turn to our attention to arrays that we can detect using CCD cameras through a technology which is called OLED, so organic LED, which is a source of light that are able to illuminate a fluorophore that can be conjugated to an antibody. By this way, we can mimic any kind of serological assay. This is a simple ELISA test where you have coated antigens, primary antibody, secondary antibody conjugated with the fluorescent, the molecule. And you can use those spotted in arrays on the surface. And by illumination and detection, you can detect a signal and have a diagnosis based on serology, miniaturized on the surface of a glass slide of the size of 1 square centimeter. And so basically the difference between a normal ELISA, which is the classical assay in the 96-well format, our that we call all-ELISA because our all-linked immunosorbent assays are actually based on chips, microchips. And we are developing in collaboration with a company here, well, in Slovenia actually, across the border, a device for doing point-of-care diagnosis of viral infection with this technology. So, tick-borne encephalitis is a flavivirus, is the most prevalent flavivirus in Europe, and spreads through all Europe from Eastern Europe, Siberia, so Russia, Siberia, towards Western Europe, including Italy and southern part of Europe. In here in color, you can see the distribution of the ticks that can carry the disease, and in red, the red dots is actually the cases, the human cases that have been so far identified in the different countries. As you can see, Italy is also partially endemic for tick-borne encephalitis, particularly in the area where we are here. So if you look at the region here is Trieste where we are, and this is the area surrounding us where we do jogging together with several of my colleagues. And we got particularly interested in this because we had, although it was well known that tick-borne encephalitis was prevalent in the Alpine area of the region, in 2013 we identified 3 cases in the Carso area, so it's just behind the institution here. And we sequenced together in collaboration with the hospital here in Trieste one of these 3 cases, and one of them is actually, they all belong to the Western European subtype as expected, and probably come from migrating wild deers from Slovenia crossing the border. So is a disease which is endemic also in Italy, and it's expanding in this case south towards the coastal areas. One of the things feature about this analysis came from a patient that unfortunately was immunocompromised when he got infected, and this allowed us to monitor the infection for at least 2 months. By this way, we could actually demonstrate that there was, as you can see here, so the IgG response was very weak because he was immunocompromised. But the shedding, for example, in the urines was sustained for almost 2 months, as you can see here, 1 month and a half, as well as the detection of a virus in blood was sustained. This does not happen normally. Usually the infection is cleared by the immune system very quickly, and the virus, you cannot detect the virus in the blood and urines. And when we fractionated the blood into the various components, we also noticed that the virus was associated with the erythrocyte fraction of the blood, well, was not found in serum plasma or in the white fraction, meaning that as it has been already demonstrated for West Nile virus, it may be a problem in case of transfusion, although the distribution of tick-borne encephalitis is more limited compared to West Nile virus, but in certain endemic area could be a problem in the transfusion field. Now, our interest is also a lot, actually, the same, principally towards virus-host interaction at the cellular level. Here is the life cycle of flaviviruses in general. You have entry and endocytosis, and you have the extrusion of the positive strand RNA, which encodes for a single polyprotein, which is then cleaved in different proteins that form both the structural components of the virus as well as the replication complex. The virus replicates at the level of the membranes, and you will see here how these membranes are organized. in the infected cell, and then will bud through the Golgi and outside in the environment. The cell respond to viral infection, as the cell always do, through the innate immunity system, shown here in very generic terms. Components of the virus, like the viral RNA, which is being replicated in the infected cell, is recognized by pattern recognition receptors like RIG-I or MDA5, which recognize the viral RNA and trigger the expression of interferon in the infected cell through IPS1, which is one of the transducer, and IF3, which is a nuclear transcription factor, which translocate into the nucleus. And interferon will then trigger an antiviral response in the surrounding uninfected cells, allowing to contain the infection. This is the basic interferon response. We demonstrated that tick-borne encephalitis is RIG-I dependent, so depends on RIG-I for its recognition. And it's also well known that viruses, in a way, escape from the innate immune sensing through different mechanisms. Here are several examples. For example, hepatitis C escapes through the protease of the virus, which targets one of these transducers, like IPS1. It cleaves the transducer, so the pathway is not active anymore. Or an S1 of influenza virus, which targets directly, for example, RIG-I. So each virus has different strategies to avoid the innate immune response. And we want to understand a little bit more about tick-borne encephalitis. So first we look at the induction of interferon of tick-borne encephalitis. So on the right you have VSV, so vesicular stomatitis virus, a different virus. You can see that as soon as the replication, so the viral RNA is starting replicating to the cell, you have also the activation of the interferon response, matching nicely the appearance of its agonist. However, in the case of tick-borne encephalitis, although the agonist, which is the viral RNA, appears quite at a normal time course following infection, the interferon response is delayed. So at 8 hours, for example, you have RNA of the virus, but you don't have interferon response. At 12 hours, the same. And the response starts at 16 hours and after that time point. So there is a window where the interferon response is not triggered by the infection. And this is an important feature because it allows the virus to propagate. So this is quite important for the pathogenesis of the virus. And so just to demonstrate that the agonist was present, we took cells, basically the infected cells, lysed the cells, took the RNA and used this RNA in uninfected cells to see if in the infected cells there were the agonist able to trigger the immune response, and it was present. So in fact, if you can use, if you have a replicating virus, you can trigger interferon. So the delay does not depend on the absence of the agonist, which is the RNA, so the agonist for the receptor, but is something else. It's something that either targets or shields this agonist from the pattern recognition receptors. It's not the case. It's not the first case, because if we transfect or we infect the viral proteins and look at the interferon response, this is not affected. So it's not a protein that is produced by the virus which actually blocks the interferon response, as happens, for example, for influenza NS1 or hepatitis C NS3, but it's something else. So we turned out to an hypothesis where actually the RNA, so the agonist, is actually shielded from the immune response by the formation of vesicles within the endoplasmic reticulum. And okay, this is a feature. Many viruses do actually change the shape of the membranes to form vesicles or kind of protective environments in the cytoplasm. And so we started to look at the geography, let's say, of the viral infection in the infected cell. This is just an immunofluorescence of different proteins, like viral proteins and viral double-strand RNA. As you can see, usually the virus replicates in the cytoplasm in areas that are enriched for PDI, which is a marker of the endoplasmic reticulum, and forms these clusters in the perinuclear area of the cytoplasm. However, the resolution of the immunofluorescence is not enough to understand how the membranes are actually modified, so we turn to electron microscopy, and in particular this is a tomographic reconstruction of an infected cell, and these are tilted images and that they were used to build a three-dimensional structure of the membranes of the infected cells. In yellow, we have the ER. These red spots are the virions which are formed, and these areas here in pale yellow are actually vesicles around 80 nanometers of size that are formed in infected cells. You may see them better here. These are the vesicles that are formed as invagination of the ER, and they actually communicate with the cytoplasm through pores. So they're actually able to communicate with the cytoplasm, but are formed as invagination of the ER. It's quite amazing. They're all very well conserved in size, around 80 nanometers, and they can form these kind of chains, one after the other, in the cisternas of the ER. When we do immunostain of these vesicles, we can actually find double-strand RNA within the vesicles, and also some viral non-structural proteins, while the membranes surrounding the vesicles are enriched with the marker of the ER, so they're actually invagination of the endoplasmic reticulum. This adds to the concept of modification of membranes in flaviviruses. These are different members, the cystic bone encephalitis, as I've shown you before. West Nile virus have the same kind of structures, so invagination of the ER with formation of large vesicles. Dengue virus also has the same structure. If you look to Hep C, in fact, it's completely different, although it's a flavivirus, so it forms extravagination of the ER surrounding replication complex. So it's the same idea. It protects the replication complex through membranes, but the mechanism is completely different. Now, one thing is to look at the formation of the vesicle and as the three-dimensional shape of the infected cells. And the other thing is to look at the dynamic of the viral RNA. To do this, we tag the viral RNA with GFP through a method known as MS2 tagging. And by this way, we are able to monitor the infection occurring in infected cells by looking at the fluorescence formed in time. We not only look at the accumulation of viral RNA in this perinuclear area of viral replication, but we can also look at the dynamic of the viral RNA through techniques such as fluorescence recovery after photobleaching or flip, fluorescence loss in photobleaching. Basically, these are techniques that deplete the signal, and by depleting the signal, you can have curves of depletion. And by this, you can actually demonstrate that within this area where the virus replicates, the viral RNA remains somehow secluded in these areas. So actually, we could, let's say, devise a model where you have the formation of vesicle in the ER, where the replication of the virus takes place, and you have an extravesicular space where the viral RNA is able to diffuse, but unable to communicate with the rest of the cytoplasm. You have to imagine that once the RNA is being translated into proteins, this protein from the replication complex then start replicating, so it converts from the positive to the negative strand and forms other positive strands RNA, which goes into this area. And this positive strand RNA has three choices. Can go back and be again translated and form again a replication complex. It can actually get encapsulated and form a nascent virion. And these aspects are controlled in this area by viral factors as well as cellular factors, and we don't know which cellular factors are involved in this pathway. So by studying cellular factors, we turned our attention to a couple of cellular proteins which are called TI1 and TAR. They are similar proteins that are involved in the stress response to stimuli from the cells. So cells that can be stressed either by viral infection or by heat or by UV or you name it. You can have a response which actually forms these granules formed of stalled mRNA and proteins in the cytoplasm, and TIA1 is one of the components of the stress response. Interestingly, when we look at the association of TIA with the viral RNA, we could see this is an immunoprecipitation of TIA and TR with different proteins and the viral RNA. So actually, TIA and TR bind the viral RNA and co-localize with the viral RNA in areas of viral replication. So this protein actually is able to access this compartment and somehow interacts with the viral RNA. In order to understand how this protein may affect the life cycle of the virus, we made a knockdown of the factors. And if you knock down either TIA or TIA1 or both, you have an increased replication in terms of plaque-forming units per ml, like as shown here. This is knocked down by siRNA. So actually, this is a kind of restriction factor for viral replication in infected cells. Finally, when we used, in this case, we used TIA knockout cells, fibroblasts, and we infected with a luciferase replicon. You can see the translation, especially early translation, heavily affected by the knockout of TIA1, indicating that possibly this factor controlled translation of the viral RNA, either early translation or also translation during replication. And so here is a complete model where you have actually several factors accessing this compartment and the formation of stress kernel following viral infection. And this may be linked to the interferon response, and we are investigating this at the moment, but I don't have data for this at the moment. I have to thank a lot of people that in the past and at the moment are joining my lab from different parts of the world, from different member states of ICGEB, and I'm grateful to all of them for the nice environment of our laboratory and the nice data that they can produce. And thanks for your attention.